Hi everyone, my name is Xiaowen Tan. Today my topic is transcriptomic comparison identifies distinct molecular features in cardiomyocytes generated by different reprogramming approaches. So a little bit background about heart disease. As the leading cause of death in the United States, there are approximately 1.5 million cases of myocardial infarction occurs annually in the United States. There are 1 billion cardiomyocytes went under apoptosis shortly after myocardial infarction, and the death of cardiomyocyte is irreversible. Due to the fact that mammalian cardiomyocytes have very limited regenerative capacity, regenerative medicine has been calling a lot of attention recently. The three main strategies of regenerative medicine is either encourage endogenous cells to regenerate, or use iPS-derived cardiomyocytes, or direct reprogram cardiac fibroblasts into cardiomyocyte. Today, we will focus on iPS-derived cardiomyocyte and direct cardiac reprogramming. So the de novo cardiomyocyte generation so far has two methods. Either went through using the 4 Yamanaka factors to induce cardiac fibroblasts into induced pluripotent stem cells, and then differentiate that into cardiomyocytes, or directly reprogram cardiac fibroblasts into induced cardiomyocyte. So those two approaches are the popular in the field, and then we want to compare whether there's any difference between the cardiomyocyte generated with two different approaches. So we will use RNA sequencing with triplicates and immunofluorescence stainings, and also future functional assessment to compare these two different cardiomyocytes. So differentiate cardiomyocyte from human-induced pluripotent stem cells has been popular for the past several years. And then there are a lot of methods have been developed using growth factors or small molecules to manipulate signaling pathways, including Wnt, nodal, TGF-beta, and such. The method that we use in this study is published in Scientific Report by me in 2019. By adding a maintenance phase in the GIWI protocol, which is shown in the schematic here. And then direct reprogramming is direct to reprogram fibroblasts into cardiomyocytes by either using transcriptional factors, microRNAs, or using epigenetic modifications. The factors that we're using in the studies are polycistronic vectors in the splicing order of MAF2C, GATA4, and TBX5, along with a combination of microRNA 133. Here is it takes 14 days to reprogram cardiac fibroblasts into human-induced cardiomyocytes. So for future presentations, HIPSC-derived cardiomyocyte will be noted as HIPSC-CMs, and then reprogrammed cardiomyocyte will be noted as HICMs. Both of the reprogrammed cardiomyocytes express cardiac-specific markers, including CTNT, CTNI, and alpha-actinin. So as we can see here, the HIPS-CMs develop spontaneously beating, as you can see in this video, but human-induced HICMs does not develop spontaneously beating. So we want to compare, the potential difference between these two. So here is the overview of transcriptomic profiles in the reprogrammed cardiomyocytes. We included H9F, which is embryonic derived fibroblast as a negative control. And from the PCA analysis with plotting PC1 and PC2, we can tell that the three cells are three different group of cells. Even iPS-CMs and ICMs, they have a lot of difference between them. So I did a cluster, I used k-means to the top 2,000 genes and then divided them into 5 clusters. And we find out that this groups here is called, it's the cardiomyocyte-specific clusters. It have a lot of gene expressions related to muscle contractions, muscle cell development, heart contractions, which are common between both HICMs and IPSCMs, but not showing in the fibroblast. And then here is the fibroblast-specific genes, including cell cycles, nuclear divisions, cell divisions, that's low in HICMs and IPSCMs. Notably, the cluster that related to cell cycles are a little bit higher in IPSCMs compared to HICMs. So what we will focus on really in the study is to analyze this comparison group, which is cluster C and D, where we have a high expressions in HICM and low expression in ICM, or inverse. So we will go into details into these groups for further presentation. So I used DEG-SIG2 to identify the differentially expressed genes, which we will note it as DEGs for future presentations, with FDR cutoff of 0.1 and a minimum fold change of 2. There are many up and downs as shown in these figures. We were focusing in the IPSCM-derived cardiomyocyte and HICMs. So by zooming in comparison of HICMs and IPSCMs, we analyzed the DEGs with gene ontology of biological process. Here is MA plot showing that there are a lot of genes that actually express differently between HICM and IPSCMs. Here, that the genes that are higher in HICMs are related to muscle contractions, cell response to chemical stimuli, and ion transport, where surprisingly, the genes that expressed higher in IPSCMs are a lot related to neuron development and cell differentiations. So that's unexpected. And we further identified the BPs that's higher in HICMs. The BP that's higher in HICMs are related to muscle system process, muscle contraction, ion transport, in phosphorate metabolic process, where genes that's higher in HIPSCMs are related to cell adhesions, cell development, and cell differentiation. And then I also run a gene set enrichment analysis in both of HICM and HICMs. And then I find out that there is a group of genes that's related to cell cycle activities that are significantly higher in IPSCMs, but not expressed much in HICMs. And then the GSEA analysis on hallmarks bring out two gene sets that's related to metabolism. One is the fatty acid metabolism, which is significantly higher in HICMs. However, the other metabolism is glycolysis, shows not much differences between HICMs and IPSCMs. As we know, maturation is always the concerns of the derived cardiomyocyte in vitro. And then studies shows that the maturations from a neonatal cardiomyocytes to adult cardiomyocytes also involves many parameters. One of them is morphology changes, and another one is the metabolism switch from fatty acid to glycolysis. Since we noticed that there is, more genes expression in fatty acid metabolism related in HICM, we want to test whether there is any difference between the maturation status between HICM and HIPSCM. So, we analyzed the maturation status using GSEA for these two cardiomyocytes and find out that HIPSCMs expressed more markers related to gene sets that's expressed in early and middle stage in heart development. However, there is no significant difference in the late and mature state of the heart development between HICM and HIPSCMs. And then because the morphology is also a very important component of the maturations, we run a cellular component comparisons between two between cardiomyocyte reprogrammed with 2 different methods, and to find out that in HICMs, the contractile fibers, myofibril contractile fiber part, and sarcomere I band are significantly higher, where we don't see any contraction related or morphology related cardiac markers showing high fold change in HIPSCMs. So in general, to sum up this study, we find out that HIPSCM and HICM are 2 types of derived cardiomyocyte with many different gene patterns, where HIPSCM exhibit higher cell cycle activities and reassemble many genes expressions fitting early and middle heart development, and showed unexpected group of gene related to neuron development, where HICM have higher expressions in sarcomere and muscle contraction related genes. So our future plan is to include endogenous human cardiomyocytes if possible for this comparison. And we will also perform many functional assessments such as Seahorse for metabolism and calcium transient and electrical physiologies for electrical properties of these 2 different cells. I would like to thank Dr. Zhang Lab and Dr. Zhou for all the helping and all the lab members in the NIH grant. Thank you, everyone. Do we have any questions?